Here, we will show you how to collect the best images from your sample using transmitted light imaging techniques such as Brightfield, phase contrast, and differential interference contrast, or DIC, and then we will move on to fluorescence imaging. In this training video, we are going to teach you the optical components and light pathways of microscopy and the differences on a basic upright versus an inverted microscope. After understanding the principles of these microscopy techniques, you will have the knowledge to use 5 of the basic microscopes available to you at IMB. This will then help you to move on to more advanced imaging systems such as the confocal, multiphoton and super resolution imaging systems. Inverted microscopes have objectives that come from the bottom of your sample, so it's useful for looking at histology slides, cells adhered to the cover slip, cells in a tissue culture dish, intact bacteria and insects and other organs in a dish. The upright microscope has objectives that come from the top, so it's useful for histology sections, slides, or cells adhered on the cover slip. For transmitted light imaging, the light source comes from the opposite side of the sample than the objective. So to attain the best images, we have to align all the optical components of illumination so that the light is focused properly on your sample. If you are performing enhanced contrast methods such as phase contrast and DIC, then you'll have to insert a phase ring or polarizer into the light path before it reaches the sample. The light that is collected by the objective then goes up through the stand to the eyepiece or detector in the camera. For fluorescence imaging, the light source comes through a fiber into the microscope stand, gets reflected by the filter cube to go down through the objective and thus is focused onto the sample. The light from the fluorophore is then reflected back up through the objective and goes through the same filter cube with an emission band specific for that fluorophore. It then goes up to the eyepiece or detector in the camera. On one of our systems, you have the option to select a color camera or a monochrome camera. A color camera converts the information to a red, green, blue or RGB image, which is good for histology slides as you can discern the staining of interest as pink and blue or brown and blue against a white background. But it makes it difficult to analyze fluorescence images. On the other hand, the monochrome camera collects the light as one color channel. Therefore, the intensity of each pixel is scaled, for example, from 0 to 255 for an 8-bit depth image.